Thank you everyone for joining. Um, let's talk about recent trends in cancer studies and some opportunities. And this is not a very long one. I again tried to keep things on a bit on the imaging side as opposed to the biology side. And so I'm going to talk about some recent trends, but more about the trends are just so many that it would be overwhelming. So I kept mostly on the opportunities side. So let's look at the way that cancer studies are done, which you have already saw yesterday, like you did. Okay,
Recording
I did not
stopped.
stop the previous recording. Hold on.
Recording in
So,
progress.
we will talk about some of the trends in not some of the trends, some of the a little traditional trends, and then some of the more opportunistic ones. Opportunistic, hopefully, can be. So let's look at the next slide. So this is a um, and so this is a looking at, so I divided it into two parts. So preclinical and a bit on the directly for the clinical. So preclinical cancer research and some of the newer things that, not newer things, but things that are happening. So I think we've covered a lot of those anyway throughout the time. So essentially, the question is that what are the recent trends in the research phases that are taking place? And do these show us any opportunities? Opportunities means like where are the gaps that are not yet covered? Is there a new technology that has not been used? That's kind of like the opportunity. But the opportunity for what? The opportunity for detection of cancer, the opportunity to use it for cancer. So, and so a lot of the, the emphasis has been on the newer ones have been really on these preclinical models, which are from mice, right? And yesterday we did three models. Remember, we did models in which tissue culture cells are injected. Again, I hope you're paying attention because that's revision for some of the questions. That tissue culture cells, human tissue culture cells, are injected into the mice in a xenograft model. Remember? And and then I said that some of the animals, okay, some of the mice already have a genetic defect. They were bred that way. They have a genetic defect, and so that genetic defect, they will produce the cancer on their own, and we don't have to give them anything artificial that way. So those were some preclinical models we had discussed. Then we had talked about an orthotopic model. Do you remember that? An orthotopic model, which was where the human cancer cells from tissue culture were injected into the organ where they belong. So if I'm studying pancreatic cancer, I would inject it into the pancreas of the mouse, not underneath the skin. So, and essentially, so that's just a revision of our preclinical models. So now these days, the genetically engineered models are more preferred. That means you have a animal, I have a mice, that already has a mutation in the gene for colorectal cancer. So if the mouse has a mutation in that gene for colorectal cancer, the mouse will develop colorectal cancer in about a couple of weeks. Because when the mice come to you, they are very young. All right, before you start the experiment, they're very young, so they will develop it in their lifetime. So and another one is, so that's a genetically engineered model. What else is a genetically engineered model? Like most of them are for the mutation that we already know. So if there was a mutation in any of the genes that we know that gives rise to the cancer, we will follow, we will do that. And it's much more complicated than that. So I just gave you a simple example of colorectal cancer mutation in the gene, and it will develop it. And then what will you do? You will give the mice the drugs by feeding it or by injecting it, and you will look at it after about a month or 2 months or so. And you will have five mice that have been given the drug and five mice that are not given the drug, and then you compare with the two cohorts to keep it simple. The next one is patient-derived xenograft. So what would be better? What would be better for a customized, personalized thing? Okay, let's see if I have a picture. Forgot. What would be nice to have a customized and a personalized xenograft model. That means there is a patient today who is, in certain stage of cancer, not in the hospital, and the patient's tumor cells are taken, and those tumor cells are then grown a little bit more into the lab, and then they are put into the mice. So if they are put into the mice, and then the mice develops the tumor, and then we can examine some of the mice with the tumors for different kinds of drugs, or targeted therapies, etc. And so those are patient-derived models, and that's fairly also a new kind of um, trend. And the very last one is an organoid. Um, so this is an organoid. An organoid is um, making a small, like an organ, like a small organ, um, making a small organ from the cells, so from the cancer cells. And what is it for? So if you have a small, all the cancer cells are in a clump, and they form a little organ, like a very small, the small size of a pea. Okay, the size of a pea, a motor, the size of a pea, and then you put it even smaller actually, and then you put it on a you bathe it with drugs, then you can see if it works or not, because then it behaves like a little tumor. So it's like a small tumor, and you have it uh, in a small fluidic chamber. I don't know if you've heard of those words. So put it in a small fluidic chamber that is the size of a capillary. So if you put it in a small fluidic chamber the size of the narrow of a capillary, then you have a blood vessel and you have a tumor inside the blood vessel. And that's what it copies. And so why are we doing this? Because we're trying to mimic or copy, okay, we're trying to copy the tumor biology, and we're trying to copy the microenvironment. Where's the microenvironment? In the animal models. The animal models give you a a tumor with a surrounding microenvironment. Okay, organoids maybe not. So in terms of preclinical, not to have too many, it's one example of preclinical that I do want to give you, and it's not that recent, is some of things from my own paper. And I have the wrong slide up. Oh, hold on. Can I just take a minute? Because slide is lost. Where did it go? Oh, okay. Sorry about that. Slide went to the wrong place. So, preclinical models, not very recent, but still xenograft related, okay, but not maybe from patient and not genetically engineered. And that example is a preclinical story of a drug called as nosh aspirin, and nosh aspirin that inhibits the growth of these pancreatic cancer cells in a mouse xenograft. And these are human pancreatic cancer cells. Now, pancreatic cancer, you know how dangerous it is. The survival, 5-year survival rate of pancreatic cancer is really not that great because by the time you catch it, by the time you know that the one person has the pancreatic cancer, it is already hard to control. And so this is about nosh aspirin. I just wanted to show you these, and actually this is my paper. This paper, I can't remember, maybe it's from 2015 or 2016. Um, or maybe 20, anyway, whatever. So I just wanted to walk you through this because otherwise there's no way to show you some of my work. And this is a protocol. It's called as a protocol. And I showed you this yesterday. Remember, I showed you that the xenograft models are just run for 30 days. And so this is a 30-day treatment where the cells, the pancreatic cells, are inoculated into the skin of the mice near the back leg. All right, and they are put inside their, they're injected in, and then for 10 days that tumor is allowed to grow. And so then the drug is fed. You literally have to take a little small, syringe and hold the mouse and feed it in the mouse. Okay, it's not does not go in the food, so it has to be fed to the mouse. And so after inoculation of the cancer cells, this drug is now fed, and it's fed, and it's only fed, either one time or depending on the dose, it can be fed more. And so then it's over 30 days, the tumors are observed. And so there's like two groups of mice or more groups of mice, depending on how many doses you have. Some of them will grow the tumor, they'll never get the drug, and some of them will get the drug. And after 30 days, you take pictures, you measure the volume of the tumors that stick out from the skin, and then you sacrifice the animal, you open up the organ, you measure the size of the organ. And I never got to show you all of that. So this is the drug is given after the 10 days of inoculation of the cancer cells. Okay, so is this a treatment model or is this a prevention model? Prevention would be that the drug is given early on. And so because the drug is given later, then this is a prevention. Sorry, this is a treatment model. So, so this is a treatment model. And so let's look at how these look. And so I just wanted to show you that these are the mice and these are the tumors that they get. So this is the control group and this is the treatment group. And so you can see that the tumors have grown so big. And in the other mice, in the treatment mice with the treatment of the drug, you can see that their tumors are fairly small. So this is the pancreatic cancer tumor that is in cell culture from some human being from some years ago. And we are using those cancer cells to test. And so this gives you an idea how this research is done. And these mice, as you can see, these mice they don't have any hair on them, right? The reason they don't have any hair on them is because it's also a property of mice that are kind of immunodeficient. They're immunodeficient, so they don't they don't reject this transplant, this xenograft. So they do not reject the xenograft, and so they are immune deficient for that reason. And so it's not the best model. It's not the best way to do it, but that's currently that's the way it is. And I just want to show it to you. Here is that after you took those pictures, etc., you euthanize them, and after you euthanize them, well, they already euthanized and then take the picture. And then you measure the volumes. So you take a vernier calipers and you measure the volumes, etc. And then when you sacrifice the animal, you look at all of its organs. Did all of its organs, are they okay? Did they have any metastasis, which in 30 days they don't? And then you harvest the tumor. And so these are the tumors. And so those are the tumors that are the control. The norm of a group of the animals, the tumor size kept on going big, whereas the one that we did the treatment, the tumor size just stayed the same. This tumor size stayed very small. Okay, so and this is just, oops, this is just to show you, I don't know why this is staying, and this is just to show you that this is from other pictures. This is just to show you that in other cases we have if you want to examine two drugs, you want to examine aspirin and you want to examine your favorite drug, nosh aspirin. This is from the market. This is an old drug and this is a new drug. Nosh aspirin is new. And when you treated did the animals the same way, you see how uh, corrosion had set in into those tumors. These are xenograft tumors. They're highly corrosed because of the aspirin has toxicity, but the nosh aspirin has very kind of like minimal toxicity here. So just to should give you an idea that you can look at organs, you're allowed to look at organs. And this one is actually, I mean, this is not exactly a xenograph, but something similar. Just wanted to give you that show. Then you done so much of, we talked about so much of immunohistochemistry, and I showed you all these complex ones. But let me show you an easy one. And so this is the control untreated with the drug. They're supposed to have big tumors, as you remember in those animals. You took those tumors and you cut sections of them, and then you did immunohistochemistry, and then you looked for whatever you want to look for. And so this is the drug that I work with. And these are so this is all from the tumors, cut the tumors, stain them, like we talked about before. And anything that is brown is kind of like taking a brown mean it is the protein is there, whatever you're looking for. So brown means protein is there. So what is brown here? Those little, so it's immunohistochemistry. We use antibodies. So it's all brown. So for this one, the protein for proliferation, which I'm not going to name here, the protein for proliferation is there. Yes. So I'm going to put it 3 checks. All of those brown there. Versus here with the drug, the cells that the protein that helps to multiply the cells, proliferation means to spread, that protein is only here, these are the tissues, but it's not here, it's not here. So for some reason, the drug really does not allow the proteins to be there. And so the protein that helps it to proliferate is not there. So this means that the proof of concept is working, that NOsh aspirin indeed works. That's the mechanism, that the cells are not proliferating. There's the cells are just not there. So actually this one just stains for the nuclei. So this one is not staining them. So next you look for cells that are You look for cells that are undergoing a, cell death. But if they're too much cell death, you will not see them. But if there are a little bit of cell death, you can find them. And so here, this is not brown. So here, it is not brown. So they did not take the antibody for the protein for cell death. So therefore, it is all bluish. And here, it is more brown. And so all of these are nucleuses that are taking the stain. So these are nucleuses that are undergoing some kind of apoptosis. Their cells are going to die. Soon they have not died. Maybe they have died all here, but these are died. They are only at a low percentage of death. So they're just about dying. Okay, these are dying cells. So just wanted to give you that example. Okay. So and you can also look in the tumors for whatever you're looking for. So if you're looking for certain proteins, certain markers, and so this is a immunofluorescence. No, sorry, this is just a fluorescence. And so this just shows you that, I'm not going to name the names, but this shows you that those proteins or that compound that you were looking for is present, and here it is not present. So that's just the immunohistochemistry to show you that certain proteins that you, because of the drug, the protein, those are there. So this is just a immunohistochemistry, and you can choose what color you want. So this was green and that was red. And with that, now we come to the talk of the day, which is advances in the preclinical models. Um, and so we're looking at, again, recent trends and opportunities. So what have we done? And so expansion of the patient-derived models. Patient-derived means cancer cells from the patient injected into the mice. Um, and so why is that done? That way, if the human being, the human cancer patient has tumors that has some heterogeneity, then it will be there in the animal. So it preserves the heterogeneity and it predicts the clinical outcome in a animal model. So that's much better than what we did uh, before. So right now it's more realistic. It's still an animal model. It's still inside the animal. It's still a mouse, but still all the cancer cells and the heterogeneous cancer cells. What are the heterogeneous? Remember, cancer stem cells, mutated cells. Was it clonal evolution? Those kind of. That's the heterogeneity we're talking about. And so this is a better model. So every time we're trying to look for a better model. So this is a better model to work with. So that's good news. And then you can use a model that is a organoid. An organoid is 3D. It's a little piece of, just like a little piece of meat, but it's really cells from cell culture put together. And they're in this microfluidic platforms that have like narrow blood vessel sized channels. And so what does it look like? Let me just draw it for you. See, it'll be this organoid. This is a beautiful model. I never got to work on that, but so this is your slide, and the slide has grooves in it. It has grooves everywhere. It has grooves just like a blood vessel. So think of blood vessel. Okay, this is not the right way to show it. So it has grooves like the thickness with the thickness of a blood vessel. So it is diameter of a blood vessel. And mm, in this blood vessel, and from here, from the side, there is a little tube. There's a tube from which there's nutrients. So nutrients are added. Nutrients are added here, and the cell is is captured in this microfluidic chamber. So, and the nutrients are coming from here, and the toxic other nutrients are exiting from here. So, nutrients are being fed. Nutrients means liquid nutrients are being fed, and the cells, you know how I said that they're stuck in a capillary? So, they're stuck in a capillary, and then they start to grow. So, they kind of like grow, and so they kind of grow and they become a organoid. So, organoids will grow. So there'll be some organoids here, there'll be some organoids here. So it's just like a little baby tumor. It's a tumor in a slide. And all you're doing is it's a tumor in a slide, and mm, and you're giving it nutrition, flow of nutrition, and you see how it works, and then you you put it in for imaging. Okay, so does it have the full micro and tumor environment of having macrophages and other cells? Not really, but it has itself, and it has its variety of heterogeneous cells. And oops. So I just wanted to show you the opportunities for imaging for these organoids. Now that I explained what the organoid is and what a patient-derived model is, and just to show you the full picture. Let me see. This has everything in it together. So here it is. So you have this is your cancer patient. This is your cancer patient, and you take the cells and you put it into the model. And so right now the thing is that what can you do with them? Well, you can try all the novel drugs. That's good. And then you can use new technologies like array technologies, and you can examine the effect. And it's really kind of like really looking for the mouse and how long the mouse will survive. Will the mouse get metastasis? Will other things happen? Can we give it the mouse, feed the mouse a drug, and will it live? How long will it live? And that is like indirectly a measure of efficiency that we can explore. And then we can take the tumors from the mice and we can keep on measuring the heterogeneity if we wanted to. So so all this tumor heterogeneity is beautifully understood, and these are just still not there to the greatest extent, but we're working on it. Things are moving. So how can you image? Okay, so our job is to talk about opportunities, right? Opportunities for not for cancer treatment, but for imaging. And so we can use there is an opportunity for this situation to apply something called as light sheet multiphoton expansion microscopy. It's a big couple of words. Okay, so multiphoton, and if you look at literature, there is like two photon and then there is three photon. So I think that's multiphoton. And what do you want to do? You want to capture. So in that patient-derived animal model, in that tumor, you want to capture the whole tumor architecture. All right. You want to see what's on the outside, what's on the inside, and you can do it by this mechanism. Okay. So, um, and here on the right-hand side, as you see, on the right-hand side, this is a organoid. Okay. So, this imaging methodology can be used for a patient-derived model or for an organoid. And I have the one that it is from this particular paper. This is a 2025 paper, and it is uh, single cell. They are able to look at in that organoid, which is this is an organoid. It has, almost looking like a tumor. So this is an organoid that was made artificially, and they can do single cell. They can, they can go and find single cell. And how are they doing it? It's by just tagging it with fluorescence, right? So tagging it with fluorescence, with either with antibodies or either with antibodies. I can't remember right now how, but either with antibodies, etc., such that certain fluorescence, certain of the dyes will only go in that fire in that organoid, and they will hexane will take up only the fibroblast. And do you remember fibroblasts are those cells that secrete the extracellular matrix? they are they're also helping it by giving it collagen, um, or whatever it needs. And here are the tumor cells. So the tumor cells in this whole organoid are this, which is here, okay, this, which is here. So they have gone in and been able to, that particular dye or that particular antibody, etc., is able to go straight into the tumor cell. And so if you, what is it, compile them together, then you get this mixed picture. Now you add to this other cells, such as endothelial cells. Now it is, the endothelial cells were probably maybe allowed to be injected a little bit, otherwise how would they get there? But the endothelial cells are green and they are on the sides and they kind of like even look like they're going to form a vessel. And so these are your endothelial cells. So this is called as spatial imaging. And so in this spatial imaging, they are able to find out all kinds of cells. Oops. They are able to find in all the tumor environment cells and the tumor cells, and what is where, who's on the outside, who's on the inside. Remember, there is glucose. Glucose is less on the oxygen is less on the inside. And so this way they are able to, this is a opportunity, and nothing has been probably done on this yet, but these people, they have been able to do it in an organoid, and hopefully this can be done in a patient-derived model. Okay, so it's called as high-resolution 3D imaging using a multiphoton uh, microscopy, expansion microscopy. So again, for organoids and these models, again, these are all preclinical, but you have to start somewhere. And so these are, we can use, one thing that can be done is, one thing that can be done is not to look for just cells, but to, this is an opportunity that one can look and perform volumetric imaging by time lapse, again, multiphoton microscopy for drug penetration. We want to follow how much of the drug has gone in, okay? And how much of the drug is going and distributing itself into the various cells. So this multiphoton microscopy can be, I'm saying, is that it's an opportunity to use it for examining, did the drug go in? If I'm giving it a drug, then does it go into each of the cells? Is it taken up? All right, so this figure that I'm showing you is not for drug tracing because we have not, this is an opportunity, it's not been done, but the technique can be used. So in this figure, which I have taken from Cell, goodness, what's the year? I have taken from a paper from Cell. It's multiphoton fluorescence microscopy for in vivo. Okay, so this is inside the animal. It's not in the organoid anymore. And what it is showing you is that it is showing you um, circulating tumor cells, CTC, circulating tumor cells that are in the lung vessels. Okay, it's not a blood vessel, but it's lung vessels before. Yeah, it must be a blood vessel. Lung vessels prior to the becoming metastasis. And so the way it's showing you, it's supposed to be the green color. Okay, I don't think I wrote that, but it's supposed to be the green color that they are following. And in this case, again, it does not, it is time lapse. It is being used, this is being used for following a substance that is inside that vessel, but it could be used for a drug as well. All right, so that's another kind of imaging opportunity for following a drug, or here in the previous one is for following heterogeneous population or the full tumor. So some more opportunity in terms of this microfluidic studies. So microfluidic studies again is a whole tumor in a microfluidic chamber. So this is the microfluidic chamber, but that's not the opportunity. The opportunity is that once we have this tumor on a chip or this tumor on a slide, then we can do some kind of imaging on it. That's the hope. So if you have a organoid or if you have a small 3D tumor that is growing, and this is a fluidic chamber where there's fluid and nutrients are given and oxygen is given in just like blood, and these this tumor, all right, this tumor is growing in it, okay, over time. If the tumor is growing in it over time, then can we use this for for taking images? So a tumor on a chip, this is really a little bit more bigger, can be this picture of this can be prepared. I mean, a tumor can be prepared by photolithography, and then you can make these tumor by bioprinting, so it can look like a tumor or it can look like a heart, and they can be applied. But that's really not it. That the opportunity for you students is really it's 3D imaging, and so that live imaging should be possible. And I don't have an answer for you how that live imaging could be there, but if you have such a small little fluidic chamber, then it should be possible to put some kind of a camera here and to be able to various kind of cameras and to be able to look at this uh, tumor, breast cancer on a chip, at this tumor as it is growing. And into this vessel, these are fluidic chambers. This is a fluidic chamber. You can introduce CAR T cells and you can also examine them and see if the CAR T cells are able to destroy the tumor. And so again, you can take it from a patient and you can use these for dynamic assessments. But the bottom line is that I'm just saying that you should be able to put in a some kind of a camera to be able to take good images in a 3D modality, which I don't know how that would be, but those are opportunities. So the next opportunities are integrating AI and computational tools in preclinical studies. Again, staying on preclinical because that's a little easier to do. And so AI applications are there in biomarker discovery. Are there any new biomarkers? So AI models can try for dose response prediction. How will it behave with a certain dose? And so there are these different kinds of mechanisms or programs to do that. I've heard of these in conferences, but these are things that if you have heard of it, then it's easy for you to pick it up. And so machine learning can help in use all of this multi-omics. What is multi-omics? Using protein, using RNA, everything, looking at it together, and analyzing it. So let's look at some. So overall opportunities again are for by using again these organoid models, that's preclinical, and it can be used for rare cancers and personalized models. So I'm just saying that um, you can use AI and the power of AI to also utilize all that data and the patient data, etc., to be able to give personalized treatment to the patients, or rare cancers for malignancies that we don't have many examples of. So you need AI models there. So recent trends is, like I said, growing the use of precision medicine. You put all that information of biomarkers, you put everything, get huge data sets, and you trying to train your data sets to see if they can predict. All right, so So it's all patient-centric approaches. Not that I can say much more here. So, how would you? If you had AI, is which is improving trial design, and AI is doing everything for you and telling you, how and what timelines are required. Then how are you going to, for treatment? How are you going to follow? And are you going to always keep looking at cells? Well, here's where these circulating tumor DNA. It seems that the tumor DNA, even if it's in small, if it's a circulating, then we can analyze it. And so circulating tumor DNA, they're released in the bloodstream. They are not inside the cell anymore. They're released in the bloodstream, and it's a small amount of DNA that is there that can be detected. Okay, and then we can use that to follow up. So we can use ctDNA. We can use AI. We're using AI, but we can follow, we can monitor with this tumor DNA. Okay, so where is the imaging opportunity again? In here, in these clinical setup, not in preclinical. So we can use something called as liquid biopsy guided imaging. Liquid biopsy guided imaging helps in monitoring. And so this is a liquid biopsy. It's just like, picking up things from the blood. And so if you can take something from the blood, like what? Like circulating tumor DNA. So if you can take some circulating tumor DNA, you're not picking up a tumor, you're just going into the blood and taking up some blood and some tumor DNA, and also some RNA, because RNA is also there. All right, what is the DNA? DNA is the genomic DNA, right? But some of it is out, some of it is out in the blood. And so then if you can take those, and then you can use it not just for monitoring, but you can use it for looking at status of disease. Then you can reduce unnecessary MRI scans, etc., etc. You can reduce all of that. So circulating tumor DNA is a small amount of DNA that can just be obtained by some kind of a blood test. However, does the circulating DNA, is it there, etc.? Is it found too much in the blood? Let me see. So, does it mean anything? That should be followed up with a PET image. So again, this is an opportunity. I just have to. Oh, I had the picture here. Um, it's a small picture. So we can combine these liquid biopsies, and we can do temporal imaging to monitor early response or early relapse. So we can try these methods so that it can be a much more sensitive way, much more personalized way of doing it. Okay. So essentially, you're just taking out blood cells and examining. Okay, we'll move on. Oh, yeah, the most important thing here is that what is this? How does the circulating tumor DNA relate with metastasis? Look at this. When metastasis increases, the ctDNA, there is more of ctDNA, and you can recognize it. So in in the blood, if there is a ctDNA that you can find, and then you need to think that micrometastasis has come in somewhere, and then that could be, you could do PET image and you can find out. Okay, so I will let this one go because it's a lot to discuss. So clinical opportunities, again, AI enabled multimodal image for trial design and response. It's a lot of words, but what we can do is, the opportunities are that with you can use multimodal imaging, such as radiology. You can fuse the radiology and the digital whole slide imaging. Okay, take the whole slide and don't put it on the microscope, but put it under a machine, and so the whole digital whole slide imaging. And you can include that circulating tumor DNA, because it's a small thing, it's a small amount circulating DNA, and include in genomics We didn't talk about genomics, so let it go. And then you can use AI to learn from there and to, to create predictive models. Okay. I don't know how that would be done, but those are the big stories that could be done. Again, these all could be done. So we can use um, we can use AI to stratify those patients based on these kind of features that we examine, etc. And so a multimodal scheme could involve PET scan image, digital pathology, a whole tile, and circulating tumor DNA. And you can feed everything into an AI model, and you can make predictions. You can make predictions of how well the treatment is going. Are there still any cancer cells left, etc., etc., in the clinic? Okay, so this will give you personalized cancer care. And that's just a big picture to explain the, the whole thing in totality. The whole thing in totality. And I'll just talk about the very top part of it here because whatever I said was the same thing, kind of. But liquid biopsy, here it is. Take a liquid biopsy, you're screening it. Again, take a liquid biopsy. Liquid biopsies, you're monitoring it. And what are you monitoring? You're monitoring the tumor DNA. Okay, I mean, the tumor is there, tumor is not there. So here, look, in early stages, there was not so much tumor DNA, but then later on, there was more tumor DNA. And then when you started treatment, the tumor DNA went less. And then when metastasis is detected, when metastasis was detected, again, the tumor DNA went up. Okay, so those are just various ways. And I think that as long as you understand that we can use imaging multimodal stuff for newer things, even for drug analysis, even if nobody's doing it, that would be that would be cutting edge. Okay, with that, I end this talk. Thank you very much, everyone.